Welcome to The Explainer. Today, we are diving into a fascinating, largely invisible science that's literally shaping the world around us. It's altering the food on our plates, the medicine in our cabinets, and even the environment we live in. We're talking, of course, about biotechnology and its applications. I'm absolutely thrilled to guide you through this because, honestly, once you see how we're reprogramming the fundamental code of life to solve some pretty massive global problems, you'll never look at a simple cotton shirt or a vial of medicine quite the same way again. So let's jump right in. To kick things off, what do the cotton in your favorite t-shirt, the life-saving insulin at a local pharmacy, and a heated international legal battle over basmati rice all have in common? Well, they're all right at the heart of the biotechnology revolution. Here's our agenda for today. First up, the pillars of biotechnology. Second, revolutionizing our agriculture. Third, medical marvels and diagnostics. Fourth, we'll step into the Transgenic Animal Lab. And finally, number 5, we'll tackle ethics, patents, and biopiracy. All right, let's get started with Section 1, the pillars of biotechnology, building the foundations. Now, what's really wild to wrap your head around here is the sheer scale. At its core, biotechnology is essentially the industrial-scale production of biopharmaceuticals and biologicals. But here is the secret. The factories doing all this heavy lifting, they aren't made of brick, mortar, or steel. The factories are living cells. By using genetically modified microbes, fungi, plants, and animals, scientists have basically turned nature's own mechanisms into miniature, highly efficient production lines. It's a real game changer. And to make these living factories actually work, the science relies on 3 critical research areas. You can think of it almost like a highly technical recipe. First, you have to provide the best catalyst, so usually a specific microbe or a pure enzyme. Second, you use chemical engineering to create the absolute optimal conditions for that catalyst to act and multiply. And third, once your biological product is cooked up, you need downstream processing technologies to purify that protein or organic compound. It's an elegant, strictly controlled 3-step dance that makes the whole shebang of modern biotech possible. Moving right along, let's see this recipe in action, out in the field. Section 2, revolutionizing our agriculture, from traditional breeding to genetic engineering. You know, the Green Revolution was incredible. It literally tripled our food supply. But even that massive leap couldn't completely keep pace with a rapidly growing human population. Traditional breeding kinda hit its limits, and the agrochemicals we relied on were getting way too expensive and, frankly, too harsh on the environment. We desperately needed a new solution, and that's where genetically modified, or GM plants, come in. Think about the specific real-world problems we can now solve. We can engineer crops to be tolerant to abiotic stresses, things like severe cold, drought, or salty soil. We make them pest-resistant, which means we can drastically cut down on chemical pesticides. GM crops help reduce post-harvest losses and actually boost how efficiently plants use minerals, stopping the soil from exhausting its fertility too quickly. And maybe the coolest part, we can enhance their nutritional value. A perfect example of this is golden rice, which is specifically enriched with vitamin A to tackle devastating nutritional deficiencies worldwide. To really visualize the impact, imagine a cotton crop absolutely devastated by bollworms, completely ruined. Now compare that to a fully mature, perfectly healthy cotton boll. How exactly do we get that healthy crop without just drenching the entire field in toxic chemicals? Well, scientists isolated a specific toxin gene, it's called the CRY gene, from a bacterium called Bacillus thuringiensis, or BT for short. They took this gene and incorporated it directly into the cotton plant's DNA. So when an insect tries to take a bite out of the plant, the naturally alkaline pH of the insect's own gut actually activates the biological toxin, eliminating the pest. The plant is essentially producing its own built-in, highly targeted biopesticide, nature fighting back. But Bt toxin definitely isn't the only tool in the modern agricultural toolkit. Let's look at another totally brilliant mechanism called RNA interference, or RNAi. Now, this is actually a natural cellular defense method found in all eukaryotic organisms. Scientists figured out how to harness this to protect tobacco plants from a really nasty root-infecting nematode. Using specific delivery vehicles called agrobacterium vectors, they introduced nematode-specific genes right into the plant. This creates double-stranded RNA that essentially acts like a mute button. It silences the specific messenger RNA of the parasite. Long story short, the nematodes simply cannot survive in the plant. The plant protects itself at a fundamental genetic level. Let's step out of the field and into the clinic. Section 3, Medical Marvels and Diagnostics, the Pharmacy of the Future. The exact same recombinant DNA technology that's protecting our cotton fields is also enabling the mass production of incredibly safe, highly effective therapeutic drugs. To put that in perspective, about 30 recombinant therapeutics are currently approved for human use worldwide, with 12 currently being marketed in India alone. A really crucial example of this evolution is insulin. Back in the day, managing adult-onset diabetes meant relying on insulin extracted from the pancreases of slaughtered cattle and pigs. And while it did work, it caused severe allergies and immune reactions in a lot of patients simply because it was a foreign animal protein. Fast forward to 1983. An American company, Eli Lilly, achieved an absolute breakthrough. They prepared 2 DNA sequences corresponding to the A and B chains of human insulin and introduced them into plasmids of E. coli bacteria. By turning E. coli into those microscopic biological factories we talked about earlier, we can now mass-produce pure human insulin safely and super efficiently. And beyond just producing drugs, biotech allows us to treat the underlying genetic code itself through gene therapy. The very first clinical case of this was way back in 1990 for a 4-year-old girl battling a severe immune disorder called ADA deficiency. When dealing with severe genetic disorders, we basically have 2 routes. First, you have the temporary fixes, things like bone marrow transplants or enzyme replacement therapy. They definitely help, but they aren't completely curative. You have to keep doing them. But then there's the holy grail, the permanent cure. If scientists can isolate the functional gene and introduce it into cells at the very early embryonic stages of life, they can permanently fix the genetic defect before the child is even born. It's truly medical sci-fi brought to life. Of course, treating a disease requires actually knowing it's there in the first place, right? Conventional diagnostics usually only work when pathogen levels are already high enough to cause noticeable symptoms. But molecular diagnostics? They've completely changed the game. Take PCR, or polymerase chain reaction. It amplifies incredibly tiny amounts of viral DNA or RNA, which allows us to detect things like HIV or specific mutations in cancer patients long before any symptoms appear. Then we have ELISA, which works on the principle of antigen-antibody interactions. It detects the actual proteins of the pathogen or the specific antibodies our bodies synthesize to fight it off. Early detection undeniably saves lives, and biotech provides us with the ultimate early warning system. Let's transition into the laboratory now for Section 4, the Transgenic Animal Lab, Biological Models, and Producers. So we're talking about animals whose DNA has been deliberately manipulated to possess and express an extra foreign gene. And the statistics here are pretty staggering. 95%. Let that sink in. Over 95% of all existing transgenic animals are mice. Now you might be wondering, why mice? Well, because they are incredibly reliable biological models. Scientists engineer these mice specifically to study how genes are regulated and how complex diseases like cancer, cystic fibrosis, and Alzheimer's actually develop. And crucially, they are used heavily to test the safety of vaccines. The polio vaccine's safety, for instance, relies extensively on testing with transgenic mice before it ever reaches human trials. But hey, it's not just mice, and it's certainly not just for testing. Animals can be producers too. Meet Rosie. Back in 1997, Rosie made history as the first transgenic cow. By introducing specific human genes into her DNA, scientists actually engineered Rosie to produce milk enriched with a human protein called alpha-lactalbumin. Churning out an impressive 2.4 grams per liter, her milk was nutritionally much more balanced for human babies than natural cow's milk. We're also using similar methods today to produce animals whose milk contains biological products intended to treat human diseases like emphysema. So again, they act as living biological factories, just like the microbes, but on a much larger scale. Which brings us to our final stop, Section 5, Ethics, Patents, and Biopiracy, Regulating the Code of Life. As we continually manipulate the very blueprints of living organisms, we naturally crash headfirst into some profound questions. I mean, we are quite literally regulating the code of life itself. And the ecological results of introducing genetically modified organisms into the wild are often unpredictable. Because of this, regulatory oversight bodies, like the Genetic Engineering Approval Committee, or GEAC, in India, have been established to impartially evaluate the validity and the safety of all GM research. But these issues extend way beyond just physical safety. They tangle deeply into economics and international law. Check out this number, 200,000. That is the estimated number of distinct varieties of rice in India alone. The agricultural history and genetic diversity there are incredibly rich, meticulously developed over thousands of years by generations of farmers and indigenous people. And unfortunately, that vast, invaluable biodiversity has led to a major modern conflict known as biopiracy. This is exactly what it sounds like. The use of bioresources by multinational companies without proper authorization and without any compensatory payment to the countries or communities of origin. A textbook example of this happened in 1997. An American company successfully secured patent rights on basmati rice through the U.S. Patent and Trademark Office. However, this supposedly new variety they claimed to have invented was actually derived directly from crossing traditional Indian farmer varieties of basmati with semi-dwarf varieties. As you can imagine, it sparked massive public anger and really highlights the incredibly complex legal landscape regarding the ownership of our planet's genetic heritage. All of which leaves us with a final, pretty profound question to chew on today. As biotechnology rapidly reshapes our world, curing diseases, securing our food supply, and fundamentally rewriting nature's limits, who truly owns the building blocks of life? It's a massive intersection of science, law, and morality, and it's a question we are going to be answering for generations to come. Thank you so much for joining me on this explainer. Keep questioning, keep learning, and I'll catch you next time.